Hello, today we're gonna talk about the Kelvin cycle. Plants undergo two types of reaction, light reaction and dark reaction. The Kelvin cycle falls into the classification of dark reactions. But this doesn't mean that it happens in the dark. What it means is that it doesn't require light. However, the Kelvin cycle requires the products made by the light reaction. So those products are NADPH and ATP. The Kelvin cycle is actually divided into three different stages. So let's start with the first one. And the first one, we have fixation of carbon dioxide into ribulose 1,5 biphosphate. And this is gonna yield 2 molecules of 3-phosphoglycerate per 1 molecule of carbon dioxide. This reaction is catalyzed by an enzyme called Rubisco. So Rubisco is the one that actually joins the CO2 and ribulose 1,5 biphosphate and breaks the unstable intermediate to form 2 molecules of 3-phosphoglycerate. Rubisco means ribulose 1,5 biphosphate carboxylase oxygenase. Carboxylase makes sense because we're fixating CO2, but what oxygenase means for this? So Rubisco not only fixates CO2, it's also able to fixate a molecule of oxygen in a process called photorespiration that I'll talk about in a different video. Okay, so let's keep going with Rubisco. Rubisco, as you see, is an important enzyme, so it needs to be regulated. One of the regulation that undergoes, it's activated by a covalent modification, meaning that the lysine residue, the 201, undergoes carbomylation to be activated. So this activation is catalyzed by another enzyme called Rubisco activase. It's also inhibited by a natural transition state analog. That inhibitor will be 2-carboxyvinyl 1-phosphate. Okay, this is for stage 1. Now let's go to stage 2. As you can see, stage 2 kind of sound familiar. It looks familiar. And it's because it's basically gluconeogenesis. And this part, we're trying to go from 3-phosphoglycerate to glyceraldehyde 3-phosphate. And in this stage, we're using the products of the light reaction, the ATP and the NADPH. And that's the difference between gluconeogenesis and the Kelvin cycle. Gluconeogenesis use NADH, but for the Kelvin cycle, we're actually using NADPH. And another interesting thing is like, it's not favorable to go this way to get glyceraldehyde 3-phosphate. However, we're able to move forward because the concentrations, the amount of NADPH and ATP, it's significantly high in the stroma, because this is where all of this is happening, the stroma of the chloroplast. So this will allow us to go to glyceraldehyde 3-phosphate. And glyceraldehyde 3-phosphate have 3 possible outcomes. So it can be exported out and be converted to sucrose, or can stay there and be converted to starch. Or the most common one, it can be recycled to generate again ribulose 1,5 biphosphate. And actually, the majority of the glyceraldehyde 3-phosphate is gonna undergo the recycling. From every 6 molecules of glyceraldehyde 3-phosphate, only 1 is gonna be used to either be sucrose or starch. But the other 5 are gonna just go to be recycled. So in this stage of the recovery of ribulose 1,5 biphosphate is stage number 3. And we still see similar pathway that we have seen before. In this red rectangle of the reaction, I'm showing another similar pathway, and it's still gluconeogenesis. And it even uses an aldolase and fructose 1-6 biphosphatase, yielding fructose 6-phosphate. And from this and forward, what we are seeing that sounds also familiar is a pentose phosphate pathway. However, it's not the entire pentose phosphate pathway, because pentose phosphate pathway had two different types of stages as well. These will be the non-oxidative reactions. And we see familiar enzymes. Let me get a closer look. So in this one, here it's barely visible, but it's transketolase, which uses the cofactor TPP. And this one, when it does, it's transfer two carbon molecules to another molecule, two carbons to another molecule. And this one, fructose phosphate, gives two carbons to glycerol 3-phosphate, forming cellulose 5-phosphate. And cellulose 5-phosphate is gonna be converted into ribulose 5-phosphate. It's gonna go back, because this is part of the pentose phosphate pathway. So the last stage, it's actually recovering the ribulose 1,5-biphosphate. So to do this, you need a kinase to donate a phosphate group, and that's how you'll be able to recover. So all of this is I don't want to explain too much, but it's like a really summary. You just are moving carbon around, modifying the molecule, so you can get ribulose 5-phosphate and use a kinase to get ribulose 1,5-biphosphate. Okay. So as the other stage, which was number 1, we regulated Rubisco. This stage also have regulation, and this is a special type of regulation because it's mediated by light. So it's not directly by light, it's indirectly actually. So what is gonna happen is that by using light driving reduction of the disulfide bonds between the cysteine residues of this type of enzymes, what it's gonna do is gonna activate all of these enzymes. And this is important because this is gonna avoid competition with glycolysis, because glycolysis is what is gonna give energy in the dark. So overall, we to yield at least one glyceride 3-phosphate to be either starch or sucrose, 3 molecules of CO2 have to be fixed. And also how this Kelvin cycle works is using a 3:2 ratio of molecules consumed. By molecules consumed, I'm referring to ATP and NADPH. So if I use 3 molecules of ATP, I'm gonna use 2 molecules of NADPH. But in this one, for that 3 molecules of CO2, I'm actually gonna use 9 molecules of ATP and 6 molecules on NADPH, because the same amount of each one of them is gonna be used here. When you fix 3 molecules of CO2, you use 6 NADPH and 6 ATP. But ATP is used also in the stage 3. That's why you require more ATP than NADPH, and it gives you this ratio. So that's everything on my part of the Kelvin cycle, and thank you very much.